So hello everybody, my name is Philippe Mercier. So I am uh, I have a formation in biology in viruses since more than 30 years and making virus bioinformatics since 25 years about. And so today I try to put a lot of resource and stuff to describe what I think is the relevant in terms of bioinformatics of arboviruses. Um, there is a lot of development right now, and even with AI. So I don't think I know every resource or tools that are now available. So if you want to share something, there's a document. I think Monique has put a link in the chat. You can put it back maybe if the chat is going too far, but there's a document you can um, put a link in there of some tools or resources that I don't speak about that could be relevant to everybody. So I invite you to share if you want to. So the course will be in 5 parts. We'll describe some webinars and meeting events at the very start, so everybody's there. After, I don't know if you'll be sleeping or gone, whatever, but then you get the information. Then introduction to arboviruses, see how their biology works and what are the problems we face in bioinformatics. Some evolution of discovery, because yeah, we have a lot to learn about arboviruses. And then viral genomics and surveillance, which are a bit the database and how you can subtype different arboviruses, so and look at phylogenetic trees, everything. And then some quick host genomics, which is very much in development, which is how we could link the genome of the mosquito, for example, with the virus, and how the virus can make the ménage à trois between the virus cycle, the vertebrate, and the mosquitoes or insects. Okay, so about the webinar and meeting, just want to point out that there's the only one I know bioinformatics meeting international will be happening in Lithuania this May. It happens every year somehow, somewhere in Europe right now. I hope we can move to elsewhere later. Also, there is a meeting in UK about, so it's not a bioinformatics meeting, it's biology, but international meeting of arboviruses, the vector. And then there are a few webinars I want to mention. You have some arbovirus biology, so it's a biology-centric webinar that you can follow. Every month or 2 months, there are some webinars about arboviruses. In terms of bioinformatics, we have the European Virus Bioinformatics Center in Vienna, which provide virus in silico lectures and viromic webinar series, which sometimes get arbovirus-centric, but at the end of the day, arboviruses are viruses, so if you make bioinformatics, could be interesting. Also, we have tools for tomorrow from the BRC, which also provide a lot of bioinformatics and AI about viruses. Okay, this is it. So now we are going for introduction to arboviruses. So yeah, arbovirus is a portmanteau name, which means for arthropod-borne virus. So it's not a real classification. There is no official classification of arbovirus. It's kind of 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 a keyword we add to some viruses, which means that they can be vectored by insects to vertebrates. Okay, but you will see that there's nothing to do with the real classification of viruses. And that's where we have a lot to learn, because we know the most prevalent arboviruses that are doing outbreaks and so on, but there is a lot to discover yet. So arboviruses are usually vectored by insects, can be ticks or mosquitoes. There's an onzootic cycle, it is a kind of natural cycle. Sometimes it can infect domestic animals, so it's a epizootic cycle. This could also infect human, but often it will be a dead end. I mean, the virus could not transmit for human. And some viruses are adapted to human and can make epidemic or European cycle infections. So yeah, this is the picture, pretty much all arbovirus replicate in the insect. And so it means they are able to replicate in vertebrate. And insect, we don't have the same antiviral system. So the virus needs to be able to face and successfully evade the two antiviral systems. There are some similarities, but many differences. Also, the virus, arbovirus, have to, it's been, for example, in mosquito eaten in the blood. You had to cross the mid-gut barrier, disseminate into the body up to salivary gland, where we could go back to the vertebrate, and then in the vertebrate, it also have to go to the blood and replicate in some organs and so on. So it's funny because it's kind of the same paradigm with the plant virus. Plant virus are pretty much all of them vectored by insects, and you have the same paradigm of the virus have to replicate in both hosts. So if you see some maps of the most important right now arbovirus like dengue, Zika, chikungunya, or West Nile, you see, for example, for Zika, that it really maps very well with the tropical region of the planet, and also arid and Mediterranean regions. I mean, these are mostly the arbovirus driven by mosquitoes, which are more active in this part. For ticks, it's more also in the north and its temperate region. But of course, the climate change is completely making things different because we have the tropical zone is going upwards, and also the loss of biodiversity is making a lot of change in the insect population, how they live, and so on. So there's a lot of perturbations in the world right now that lead us to fear that so some arbovirus might emerge or change the way they infect vertebrates or humans. We've seen that already with Zika virus recently, because originally it was known in Africa and it went all over the world. Also, chikungunya virus made some epidemics and continues to do so. So it's really a topic that we know in the future will have more and more importance, especially in the center of the planet. The good news is that many researchers on bioinformatics are leading the charge in Africa, South America, and Asia, which are the places where they are most prevalent, and it's really a very good thing that we have leading bioinformatics in these countries. So I told you arbovirus is not really a classification, it's a kind of nickname. But the classification of arbovirus, we see just impact few viral families actually that are able to infect both vertebrate and human. Most of them are RNA viruses. So you have, for example, alpha virus with chikungunya, the flavivirus, yellow fever, Zika, and so on. You have a few rhabdovirus, which mostly infect actually animals, and very few like dead host to humans. The Fenui virus and also the Bunya virus are doing a lot of problems into the world. But by looking, so if you look at the description of arbovirus, you will have many different lists in internet. Some will just put dengue, flavivirus, and the Zika, chikungunya, and so on. And some will give you a list much more diverse. So here I'll try to focus on everything that can go up to, you see, few DNA virus actually are arboviruses, not only RNA, but they don't make epidemics or things in human. You have ASFAR, which is ASFAR virus are infecting pigs actually, and it's also arbovirus. And recently, I have found by looking at ICTV, so the International Taxonomy of Viruses, they provide a list of viruses with their host, and I found that two of them in this list infect also vertebrates and insects. And it turned out that these are kind of new, that the parvovirus and endovirus and densovirus. This is publication, I think, from Brazil, which identified that. So actually, the number of arbovirus in the world are kind of underestimated. We know the main part, I mean, the mixed epidemics of yellow fever, Zika, and so on, which is the most important one. But in terms of discovery and surveillance, there's a lot to learn. So if you look at the main arbovirus, known arbovirus, it's in the RNA viruses, and you have a few double-strand RNA viruses in the real virus. Also, yeah, here in the positive strand RNA virus and negative strand RNA virus have a lot as well. But I think these ones are doing the worst because you see the flavivirus, for example, there are many of them. So here I put the name of some viruses, but it's not at all exhaustive. Also, there's a kind of problem that people name the virus when they discover an epidemic on the sequence. And sometimes it's kind of the same virus genetically, but they have different names, or they are same serol group. It means that they are different continents, but they are doing the same disease, and they can neutralize it for each other by immunity. So it's kind of difficult to know how many of them are known because you have a lot of ways to name the same virus sometimes. So it's a bit a problem, but we have some official classifications. In ViralZone, there is a human virus table in which you can, so this is you go in ViralZone data on human virus from here, and you can type arbovirus and you will have a list. I think it's 123, but this it's only this number because I have grouped that by kind of serol group on the virus that do pretty much the same. For example, the EIAC virus is present in North America and Europe, but Colorado tick fever just in North America. But these are pretty much doing the same on the California hair virus as well. So in this, I try to put a lot of different features like the tropism of the cell infected, which is very important but difficult to find in a coherent list and in database. And here I use the ontology, the Ubero ontology for the organs and the cell ontology for the cells. So it requires a lot of research in papers to find this, and the work is not finished yet. But you have for every virus at least some kind of so sometimes the incidence or prevalence of it, the geography for different viruses, the transmission, which is a arbovirus. You see, it's kind of used as a transmission name because that's what it means actually. The tropism of cell and the pathology links to Wikipedia, the disease pages actually. And sometimes the fatality rate, you see that it still doesn't kill people, hopefully, and links to some genome assembly and so on. So by doing some search on this smart table, you can find the things you want. Also, I told you about international taxonomy of viruses, so that the official taxonomy classification, there really is a new taxonomy every year. And they are providing a very nice document, which is called the virus metadata resource, which lists all the official species, okay? So now, if you look at the one which infect invertebrates and other hosts, you have a lot of them. I think it's 533, which will be arboviruses. Okay, these are official species which you could classify as arboviruses because they infect invertebrates and vertebrates. So as I told you, the number can vary depending on the name you give them. But so these species sometimes are very close to each other or doing the same disease, okay? So this table also present on ViralZone. This is a copy of ICTV, which provides an Excel file. It's not very easy to work with, and you can, by the search, find the data you're interested in, and if you export to CSV, only the data you have selected will be exported, and then you can work with it and do whatever research. You have, at the end of the table, links to genomic sequence. Here you have gene bank links. So you can also have directly into sequence from this representative for every species designed by ICTV. So noteworthy, there is an effort made by people in Asia that was published in Nature Data with a global data set of sequence for arboviruses. So they use, as I told you, ICTV, but many other things, gene bank and BL, and try to find out through the host and all the data to gather all the sequence, all the genomic sequence of these viruses. So they end up with a lot of sequence, like 100,000 sequences. And they made a data set that can be accessed to Figshare. So the problem is that it's like 3 years ago it was published, so I don't think it was updated yet. But you can see here on the map all the genome sequence they get in this data set. In red, it's for mosquitoes. In green, it's for ticks. So you see ticks are going farther north than mosquitoes, actually. So this data set has merit to exist, and they try to make a sort of exhaustive picture of the arbovirus genome that we have in all databases. So if you look at the figure in this paper, you see what is called of arbovirus or arthropods specific. It means that these are insect virus not able to infect vertebrates, and these are arboviruses, okay? So you see a lot of families, and this represents the number of what they have. And you have many families that are not even present in the ViralZone part I showed you because they're found association from different databases. So that's what I told you. It's difficult to get the whole picture because it's all over the place. Okay. Now we're going to talk a bit more about evolution and discovery of these arboviruses. So, so one thing I like to consider is how this virus evolved actually on the planet. So, I mean, land viruses have faced a big problem in history. When life conquests land, viruses had a problem of transmission because transmission is water is very efficient. You diffuse passively into the water, and most organisms don't have very thick skin. But when you go into the land, it's a much different system. So, for example, the first to colonize land were hopefully been plants and fungi. And fungi have a lot of specific viruses, but they can transmit for each other without going out of fungi through rhizome, actually. So they are kind of very niche viruses, and they don't transmit very well to plants. But virus of alga has a lot of problem to get into plants because plants don't move, there is no water, and there are a very thick wall around the cell. So really, really bad for viruses to infect and to make the jump. But what happens is that insects emerge also on land, and these are really perfect hosts for viruses because they are very numerous, they reproduce fast, they don't live long, so they don't have long-lasting immunity, and they interact with the plants and even with the fungi, so the virus could then in history start to conquer land, hopefully because of insect. And then when you get the vertebrate, also you have exchange between vertebrate and insect, not between plants and vertebrates, because these viruses are really, it's a big jump. They don't jump from one to the other. So the insects represent a very huge importance still in the virus dissemination in the planet. And actually, if you look at today, there are more than 1 million species of insects, and it's considered to 10 to the 30 number of insects on the planet. It's an estimation, of course, where only 30,000 vertebrate species, and with about 10 to the 18 number of vertebrates. So we have much, much more insects. They reproduce faster. They have a lot of viruses. So they are still a reservoir very active of virus transmission in the planet and on land, actually, because I'm just talking about that. So the potential for arbovirus is very strong. Actually, we are lucky that is not so many doing epidemics. But with global warming and biodiversity collapse, we can fear that this equilibrium we have right now could be different in the next years. Okay, so if you look at the insect viruses, I showed you before on ViralZone what you have arbovirus. And now you see just insect viruses. You see it's all over the place. Every class of viruses are infecting a lot of insects. And there is a bias in the data right now because historically we have sequenced first the human viruses and also the domestic animals viruses and plants. And now we start with metagenomic to sequence more and more insects, and we discover a lot of them. So it's just a bit view from far away of the page, and you see that negative strand RNA viruses, there's a lot of insect viruses there, many discovered by metagenomics. So we have still biased data, but it's going a bit better thanks to metagenomics. But you will see that we need some tools to make sense a bit more of this metagenomics. So for example, this is a publication in 2020 of people that use RNA-seq analysis to find RNA virus in insect, and you see in red that the new virus insect they found. And you see for the picornard, there are a huge number of insect virus. For the flavi, some of them, the partiti, and so on. So you have many virus families that we didn't have many insect hosts before, turn out to be mostly insect viruses. So this can suppose that some of these families were originated in insects. For example, pox virus, which are DNA, are presumably originated in insects and then get to vertebrates. Few of them don't, like astrovirus, you see they didn't find much insect or nido, like SARS-CoV-2 is from nido, for example. They don't really have links to insects right now from this study of the alpha virus also, like chikungunya, doesn't have links. But many other families are really a huge link to insect. So this is just one study, and already it kind of revolutionated what we know about viruses, and it's still not yet classified because it takes a lot of time to identify from metagenomics. Okay, now a case study which will show you some interesting stuff. It's a blood analysis for Tanzanian children that were done in collaboration between Africa and Switzerland a few years ago. So this is a publication, the blood virosphere. So actually children having febrile being sometimes ill by some viruses where their blood were collected, and we look at the virus that will be there because it was children sick without a real diagnostic. We didn't know what the pathogen have. And they make metagenomics on all the blood samples and find like many RNA virus of clinical significance, like hepatovirus, I don't know, Norwalk, rhinovirus. So this is kind of normal, okay, we know we get it. But also some of unknown significance, which are kind of weird viruses that we found. And if you look at it, for example, there is a lot, 12 patients we have found disease-true viruses, which are insect only from what we know. Also, 24% have tetraviruses, which are also insect only from what we know in taxonomy. So this work suggests that maybe this may be hidden arboviruses that we never saw before, but are just happening in Tanzania, I think, or maybe elsewhere in the world. And if you look at the literature, for example, this disease-true virus have been also seen in wet squirrel and so picornavirus. So there are many publications that suggest that there are more kind of arbovirus at the least we know yet. And this kind of study can highlight it. The problem is that it's difficult to know if it's really an arbovirus because you detect that in the blood, okay, but you don't have experimental or evidence that it was transmitted from insect and replicated also in humans. So it's just a hint, but we need some tools to go further. Another discovery, for example, that was made is a Tanzavirus from kind of the same data. They isolated a new flabovirus. You see, this is phylogenetic of flaboviruses. This is one is clearly different, and it links to a new genera actually classified by CTV. This is the ViralZone page of the Tanzavirus, so which is accepted to be as an arbovirus. So you see, it's kind of happening to use metagenomics to find the new arbovirus and to describe even more the landscape of it. But the big problem is that when you make metagenomics, how do you know about the host? So we need some tools to predict the host of these viruses, hopefully an alignment-free method, because if you align the many viruses will come together even if with different hosts. And for that, there is people are using codon usage, because it turned out that during in insect and human, for example, codon usage is different. You have some avoidance of CPG or an insect of APU, I think. And by studying this, you can have an idea of it. So you see, so from some viruses, you look at the codon usage, and you see that many families have different codon usage, which does not always correspond to the host. So it's kind of interesting on a way to go, but the problem is that pretty much each viral family have a different perspective on this codon usage. Some, they don't care and they don't follow the host. Some follow the host. So it's kind of complicated to do, but it's still a good way to check it. And so I found Mercier, which is made by AI. Actually, it doesn't focus on codons, but on many features, on which is what actually we need to. I mean, hopefully we have at the end of the day an AI that will be able to tell us if a virus sequence from metagenomics will be able to replicate in vertebrate or not. And this is what they do for mosquito. This is a publication. It's from South America. And what they do, you insert viral counting FASTA, so you have to have isolated the virus countings. Then they extract what are other viruses compared to mosquito-associated viruses. So they kind of put aside any virus that will not be able to replicate in mosquito. And then they will split between arboviruses, so able to replicate in vertebrate and mosquito, or only mosquito viruses. And it can go down to 500 base pairs. At the end of the day, you have an estimation. For example, this, you have 3 contigs, and you see that this one is not infecting mosquito, hopefully. This one will infect only mosquito, but not be an arbovirus, considering the result. And this one is likely to be an arbovirus. So that's really the kind of thing we need. And Mercier, I think, is a very good way to go. What I see a bit of problem we have is that the training is based on current annotation, and you see that you don't have much insect-specific in blue viruses in many families. So we have a gap of knowledge, and I think it works very well, but it will get even better when the gap of knowledge will be more filled. I mean, we have more data on insect viruses. So to predict the virus insect vertebrates host, AI, I think, will be the key. But it requires maybe more unbiased data that we have right now. Still, what we have now, like to see with Mercier, is actually working. Just could improve with better data. Okay, what we can do also is when you got a virus by metagenomics or whatever way, is to annotate it by alignment-free method, because virus, especially RNA virus, they diverge a lot, and sometimes you don't get any function of the proteins you get in the genome of an arbovirus. So for example, it's a work I did with the Colorado tick fever virus and many other retroviruses. You have the genome is 12 segments, and only one segment is known. A function is known. It's a polymerase, because polymerase, you have an active site and you identify very quickly. Others, they don't get anything, even by Interpro, PFAM, everything that you could find some domain, they don't find a clue. So after sequencing a long time ago, actually, you don't know what these proteins do. But there is a way now with AlphaFold to check it, and actually it works pretty well for Colorado tick fever virus. So I modeled every proteins, 12, it's not so much actually. And you have the polymerase that came out with a good score. We expected that, the polymerase is polymerase, but then we found other proteins have very good score, and then we found the capsid proteins on the capping enzymes on the outer capsid. So it was possible to identify the function and to predict based only on an alignment-free method by just predicting the structure with AlphaFold, which I think for arboviruses will be pretty nice. I mean, for every viruses, but especially these viruses. For example, so VP2, we have a fold with a good score. You see in blue is a high score of PLDDT, above 90%. So it means that we have 90% confidence that this structure is real. And when you use VP2, then you use the structure made by AlphaFold, it's AlphaFold 3, to check for the identical structure using FoldSeq. So FoldSeq is the structure comparison tool developed in South Korea by Stanager Lab, and it's extremely efficient and fast. And then I put this structure, and then I get a lot of results. So you see description is not real because it's a RDRP context, but it contains the capsid in the PDB. So I've checked with this PDB to have experimental structure, and then I can say that it's very identical to the capsid of SIPO viruses, which are close genus. But if you look at the result, you see that the sequence between the SIPO virus and the Colorado tick fever is not conserved at all. You have nothing. So I told you it's a alignment-free method because they don't align, but the structure is extremely the same. So it's a good tool to go beyond the problem of sequence diversity. And yeah, AlphaFold 3 did better for these viruses. You see the difference between AlphaFold 2. This is on the side of the capsid, which is kind of very thin. But so this is an experimental PDB. On the right, in the middle, you have AlphaFold 3, and in the left, you have AlphaFold 2 results. You see it was not seen at all in AlphaFold 2. So we have a better result with AlphaFold 3. Most of the time, actually, but in some cases, high quality structure made by AlphaFold 2 can be better than 3. I don't know why. But so if AlphaFold 3 fails, you could look at what is in the database with AlphaFold 2 as well. So it's the same with the capping enzyme. And here, even the enzymatic activities, there are 3 enzymatic activities. They were not detected by PFAM, Interpro, or whatever, by the sequence, but the structure, you see, it's completely clear. You can find the active site, everything. So it's very powerful, and we're able to identify the function of 4 genes out of these 12, thanks to just structural modeling. Okay. So I will talk about some database like ENSDC, which is GeneBank and Trends, Pathoplexus, Loculus, BVBRC, also ARCA and RVIR-END, and some tools like NextTrend, NextClade, Genome Detective, which are some very common tools to use for subtyping or looking at phylogeny of viruses. Okay. So INSDC is the central database for sequences. So it's a consortium between NCBI, DDBG in Japan, and European Nucleotide Archive in England. NCBI is in the US. And that's the main database for all sequence overall. I think every people, you know it. And that's where all the sequence we share in the world should end up. So, but it's not well designed for viruses. It was made previously, it was made in the first place for cellular organisms. Okay, well, they get all organisms, including viruses. But with viruses, we need a lot of metadata. So geographical location, the date, and a lot of metadata is important to make sense of our sequence that NSDC is not doing or will not do, at least in the coming years, because it's not in their plan. So we need specialized database to take care of virus sequence. So one of them, famous, for example, is GZ, but GZ is not open access, so I will not speak about it. Also, GZ is not sharing their public data to in SDC. So they take from NSDC, they get sequence, which are public in their database, but they don't share in NSDC. So, and it's a private foundation, so we don't, it's not the thing I want to put into perspective. I will show you other possibilities to share actually and study viral sequences. So one which was developed in Switzerland recently is called Pathoplexus. So it's kind of similar to what does GZ with viruses, but it's open source, it's community governed platform. You see that already they got some arboviruses with Chlamydia Tongo and West Nile virus. So communities are starting to deposit sequence and they're starting to create a workflow for this. So Pathoplexus is empowered by researchers and it's driven by Loculus, which is the software behind it, which is very interesting because Loculus actually so it's driving the database. I will show you a bit like what is the output, but it's also open source and you can use it to have your own database, even private database in your institute, in your university, in your lab, whatever. So Loculus is completely open, so you can profit all the features for your private data if you need for, I don't know, for vaccine design or publication. Or you can also share on the public Pathoplexus using the same. So that's what you have, for example, when you look in Pathoplexus and West Nile virus, you have all the sequence deposited, and it's the same team that work on NextTrain, so it's very easy to use. There are a lot of connection with it, and you can check the sequence even by mutation, which is good. So, for example, you're looking for one mutation so that you know maybe would make a drug resistance or an antibody binding resistance. And you can file the sequence very quickly by just typing there. So you have a lot of feature. It's still in development, I think. It's starting, but I see a good future, especially because it's completely open access and reusable and exchange everything with GenBank that they have. Of course, you can submit and keep the sequence private for some times for publication, but then what is shared in Pathoplexus is shared in GenBank as well. So the software is called Loculus. You can access to Loculus on the Loculus site. You can download it. You have a lot of explanation how to install that and use that if you want to use for yourself for your studies. So I think it's a good thing that everything is open access there. There is also a specialized database in the US called BVBRC virus, which was before called Viper. And they have a lot of families to which arbovirus belongs. So you see, this is a homepage of BVBRC virus. You have Bunia virus, Flaviviridae, Toga, and so on. And some feature virus like dengue and Zika. So you see, I told you every time you look at arboviruses, you have kind of different names. Nobody has all of them, but you find some good stuff there. So what allows BVBRC is easy genome browsing. So you have an overview of all the genome with their host. So they make a focus on metadata. So you can see the countries, collection year, and so on. And you got a lot of tabs who taxonomy, genome, feature, protein, even epitopes from some viruses, which could be relevant for immunity, vaccines, monoclonal antibodies. And that's what you get, for example, from, I don't know, I use dengue. So you get a list you can download, and you have a lot of tools on BVBRC, bioinformatic tools, to blast, design primer, align, and so on. So it's really rich environment, and there are given workshops to learn how to use their stuff. There are videos also, so you can check on their site if you're interested. Also, we have ARCA, which is the Arboviruses reported case in the America, which is a database specifically for Americas. There are a lot of sequences that can be interesting to go look at if you want to. You see you have chikungunya, dengue, Zika, so only these three ones, which are the main problem of epidemics. I found also a database in India, which is RVRand, which people are putting their sequences. So you see that the fact that we don't have a good big database for arboviruses, a lot of people are making a small database to share between them, which is good. But then you have to go in different places to get the whole picture. But that's really actually dynamic, and it's very good to see all this data shared between people, because at the end of the day, it's not a big problem to transfer one to each other. So this is arboviral proteins for Indian subcontinent, which is specifically dedicated to arbovirus, also this one. So nice to know. So okay, now about more tools on visualization for surveillance. I will show you NextTrain, which is widely used and extremely efficient, completely open access as well, to look at the phylogeny. So if you go into NextTrain, there are a lot of workflows for different viruses. And you have also a different set of arboviruses. So you have chikungunya, dengue, but also Oropouche, West Nile, yellow fever, and Zika. I will show you later, but you can create community workflows on different viruses if you want, because everything is open and you can access to all the software and communicate with them if you want. So NextTrain is part of WAL. It used to get data for GZ, but I think that recently GZ start to cut this because they don't want to share anymore for whatever reason. But they share with GeneBank and LocalFasta, so they don't really receive sequence. They just use sequence for public databases. Okay, so they have a module called Augur, which make the analysis. So put all the sequence together, compare them, classified with the metadata, put into a relevant JSON file, I think. That will be visualized by the Auspice module. So Auspice is for visualization of the data, the phylogenetic trees, the place on the planet, everything. So if you have a different way to produce these files, you can look at it by Auspice, because Augur Auspice also open access. You can get it, you can use it locally, or you can use it through NextTrain. But NextTrain will get only sequence from this part. So I show you, this is a file which is made by Augur from all the sequence. So it just calculated the trees and all the metadata, and Auspice will make a look, a pretty nice look actually you have in NextTrain of the phylogenetic trees, so you can a bit make sense of the data. On the site of NextTrain, you have a phylogeny panel. You can check and decide if you want some dates and some place on the world and how the tree is displayed here, different type of trees. You have, of course, the tree with different colors for the subtypes. You have a map. So here it's a Zika virus on NextTrain. So you have a map. You see where Zika is more prevalent, and the different strains corresponding, the color corresponds to the phylogenetic tree. And you have here the kind of heat map of mutation or variability on the genome of Zika, which is a flavivirus. So you see there's a lot of variability actually happening there. But then if you want to make sense, you have to look at which part of the tree you're looking at, because here it's a whole tree, okay? So in the phylogeny panel, I told you can check the date range or something. In the resulting phylogeny, you can hover and click and get information on this part of the tree, which is very nice actually. You see the different countries, you see, so all the metadata are available in the different perspective, which is really the power of this kind of feature in Xtrain. Also, when you click on the diversity, you get the mutations. For example, here on this virus, if you click there, so you see a few mutations on it, but you highlight the part of the tree that contains a mutation actually. So it can also help you figure out what is happening in new sequence in top of the phylogenetic trees, because nowadays you got many sequences in phylogenetic trees, so you cannot really look by hand at it. You need these tools. So in Nextstream, there are some builds, which are the recipe of code for some viruses. So I showed you that there are Arboviruses, Zika, and so on. There are different builds that will take the sequence from that public database, make all the work, and then you have the look in auspice. Nextstream is easy to run when new sequence is available. So if you want to make it locally, so everything is described how to use it on all the details in Nextstream. So you can use your own lab data or your data plus the public repository. You have all the range of possibilities to work with that. So in summary, it's a powerful tool to look at phylogeny and look at what is happening actually, different circulating subtype and so on. It was widely used in coronavirus pandemic, for example. It's a real-time snapshot, so it's updated very often. There's an interactive web application, Auspice, that you can also use for yourself, which is pretty good. And a lot of documentations and tutorials at nexttrend.org. So if you want to use it and to make this same of tree and use this kind of software to survey whatever virus you want with whatever sequence, you can look at documentation and use it. So there are, of course, many other ways to view. So Nexttrend is one of the most famous, but as an example, I show you MicroReact, which is made from people in Europe, which is a bit the same, actually. They will display the result of alignment of sequence with a tree and dot on the planet. So it's a visualization. It's a bit like Auspice, actually, from Nexttrend. Also, it's open access, and you can get it and use it how you prefer. So there are very good solutions made to study the real-time phylogeny of arboviruses and have a clean view of the output. Now, one very important thing is to subtype arboviruses, because arboviruses tend to genetically drift, or you have different subtypes already circulating, like dengue 1, 2, 3, 4. And when you get a sequence, one of the most urgent thing to do is to know the subtype of it. So you know where you are. If there's an outbreak with this subtype and you get it, so, I mean, okay, it's an outbreak getting on, but if you have a new subtypes, then it may be a virus that came from elsewhere or something happening. So for subtyping, I will show you about Nextclade, which is actually doing by the same group of Swiss-Prot, Nexttrend, sorry. We show you also genome detective tools, which are there since a long time and providing very strong subtyping tools. And we tell you about the future, which is AI-driven genotyping, subtyping. So Nextclade, I told you, is brought you by the same group. in Basel. And yeah, it's open source project, always open source, which make alignment, mutation calling, and clade assessment, so subtyping, quality check, and phylogenetic of your sequence. So you submit your field sequence to Nextclade, and they will tell you which are the subtypes, mutation, and so it will show you. So when you go into Nextclade, that's what you get. In the left box, you provide your sequence data in FASTA. So everything that you make an assembly from genomic sequencing, and you want to check for these viruses, for their clade, their sequence mutations. You put the whole into FASTA there, and then you choose a data set. Okay, so Nexttrend to work with, you need to have a reference data set. So I showed you the arboviral dataset present in Nexttrade. So made by the Nexttrade group, you have dengue for all serotypes and yellow fever. But you see that you have community driven because it's a feature you can by community and contacting Nexttrade make a sequence dataset, which will allow you then to look at your favorite virus. So you see a lot of community driven have been made by for arboviruses, which is great. So you have a Crimea Congo, chikungunya, and all dengue separated and not with all serotypes together. Okay, here you see in the reference, they give you the reference actually sequence, which is nice because then you can really pick it up and use it if you want from different perspective of yourself. So it's a way to get a reference that is already used and selected for subtyping, for example. And yeah, you see the last update and dataset name and so on. So you check your dataset, you put your sequence, and Nextclade, sorry, will give you some results. So these are actually COVID results I was making during surveillance in Switzerland, but it's the same for all viruses. You get the clade, the lineage, you get the variants. For example, if you look, if you hover to the variant part, it will tell you which amino acid or nucleotide change you have. Here you have a deletion and you have few things. So for each sequence, you have access to an abstract of what is relevant to this sequence compared to the reference. I showed you before, every dataset has a reference. So when you compare to the reference, you get these mutations. So Nextclade can give you a lot of output files with the data it produced. So it's very convenient. It was also used a lot during COVID, for example. But you see for arboviruses, there's still a lot to do. But again, we have like 1, 2, 3, 4 viruses, 4 arboviruses only, which is not the whole thing. Now I will present you a genome detective, which is made by collaboration, I think, between England and South Africa. Tullio is very, the Oliver is very active in bioinformatics in Africa, and the whole Africa is producing a lot of tools now, especially for arboviruses. So Genome Detective is one of them, and Genome Detective is a kind of bag in which you have a lot of tools on very nice bioinformatic stuff. Very well done, and it's done since many years. I mean, subtypings was already there like 15 years ago, so they have a lot of experience. It's not like a new thing made like this. For example, if you look at their subtyping tools, you see they have chikungunya, dengue, what else? They got ropush, West Nile, yellow fever, Zika. So already a good set of subtyping tools for the main arboviruses circulating, in which you actually enter your sequence and they will give you a subtype. So as I told you, there is a site in South Africa from a KwaZulu-Natal Research Innovation Sequencing Platform, which gives you access to the subtyping tools, the genome detective part, which I did not mention, but genome detective also is a way to to identify virus in metagenomics. So that's one of the main part of genome detective. You can put some of your raw data for metagenomics in it, and it will make some assembly, and it will detect from reference database some viruses. Everything is automatic by your browser. So it's pretty efficient to make a first run of metagenomic data for viruses. And in plus, they got the site tapping tools. Okay. So if you're interested in this, you can look, just type the detective, and you will see there are also everything explained on the site for all the tools that are present. Okay. And here also you have several bioinformatic tools, including Genome Detective. Now I told you that the future could be AI classification of subtyping, so, sort of. And there is also made by the Tulio group in South Africa. The first one I saw, it's CRAFT. It's a chaos random forest system to dengue subtyping. And it's made by a consortium of Inform Africa. And actually, they succeeded completely. So they made, using AI, all the data we have on some types, we have many, many sequences. You know, with AI, you need a lot of data for training. The more you have, the better the training is. But for dengue, we have a lot. For would be possible for Zika, West Nile, and so on. So here for dengue, they show that it's very accurate, but especially it's very, very fast. So they have really good accuracy on the test, but 140,000 sequences per minute, which is crazy. Most people don't sequence that much, actually. So so it's a big improvement in efficiency that will be useful when you have really huge data, actually. But I really like to see that advance toward this, because at the end of the day, having fast tools and accurate is always better. So Craft is there. You have the source code available in GitHub, and you have the paper, which was published recently, you see, 2025. And I suspect that people will develop a bit more on other arboviruses, subtyping tools using AI in the future. Okay, another tool that is worth mentioning is Glue for arboviruses. So it's made in Glasgow, in the Center for Virus Research. It's provided them, and it's a data-centric bioinformatic environment for sequence virus data. It's focused on variation, evolution, and sequence interpretation. So again, many people are sharing sequence with Glue, and you can download it. You see download Glue here on our page. And there is many, how to say that, many tools to study the diversity using Glue. You see, you have a whole landscape of tools already present. And also, I discovered recently Galaxy, which is not dedicated to viruses, actually, or arboviruses, but it's worth mentioning. It's open source, and it's made in America. But actually, it's web-based, so you have no code interface, and you can do a lot of stuff from bioinformatics, from metagenomics. Even go to Nexttrain, you will see, with different workflows and with the no-code interface. So it's kind of easier interface called Galaxy, and it's also a group of many tools which are shareable and reproducible. So it's a good way to have things like that put together different systems. But it's not dedicated to viruses. So you have some things which are relevant to us and others maybe not, okay? So for example, with Galaxy, you can make metagenomic detection, identify reads for mosquito pools using Kraken Diamond. You can still look at quasi-species with IVAR low-freq. You can send that to next-train augur for then look into phylogeny, for example. So you see, it's really a place that seems to allow to use many different bioinformatics tools together without much code, which can be nice for many people, actually. Okay, so now we are going into the last part, which are host genomics. So here we are more in the research than really actual set of things, because it's just kind of starting, okay? So the arboviruses, they are specific. They can infect actually many vertebrates often, not really much restricted in terms of vertebrate, but for insects, they are really specific to the vector. So for example, I took the list of 123 human arboviruses in my list in ViralZone, which is there. And I ask an AI to classify by the different hosts I have in this data. And you see the result is that you have many mosquitoes, few ticks, and sandflies. But there is no kind of logic compared to the classification of viruses, because you have flaviviride that infect many different hosts and so on. But each virus will be kind of specifically transmitted by this host. So you have some by Aedes, some by Anopheles, sandflies, ticks, and it determines, actually what will happen in the geography relative to the climate and the dynamic of epidemics also. So it's very important to know of the host. And often, you get some things in gene bank and so on, but the data is not really perfect to know exactly which virus use which insect specifically. So we have a few data, but it's not completely put in place that you can really work with using AI, for example. So I use this, but it's not exhaustive what I have in this table for these various groups, okay? It gives you hints of what's happening. Also, yeah, it's a ménage à trois. You have the virus, you have the vertebrate host, you have the insect host, and the virus needs to infect the cells in the insect and the vertebrate. They need to find receptors to enter these cells. And which receptor are there? It's a big question. So there is a viral receptor table in ViralZone in which I put everything when I came across a publication. Again, it's not exhaustive, but it's still there. And you see, for example, I just type dengue in the smart table. So you have a lot of dengue receptors, like it was 15 different receptors recorded for dengue. And one of very important thing is that you have the type of receptor, if it's adhesion or entry receptor, because viruses sometimes can adhere to cell using a receptor, but they will not enter. Some receptor will allow you to enter the cell. Sometimes they will adhere first to some molecules and then find the receptor of entry. So adhesion receptor is not the same as entry, but it's still important. And also, I look at the expression in the cells because when you have a receptor published that it's a receptor of virus, you expect this receptor to be expressed in the cells the virus will infect. And it's not always the case, okay? But here, for example, you see in red that this mannose receptor is expressed in macrophage, and actually the virus, the dengue, is infecting macrophage dendritic cells. So there's a logic here. If you don't have this logic, it means you're not sure that the receptor is really working in nature. But these are pretty much all vertebrate receptors. I don't find many data about insect. Okay, so I found this review of insight into mosquito-borne arbovirus receptors. It seemed that they described some common receptor, vertebrate and insects. Some proteins are common and conserve enough between insect and vertebrate, that the virus can use the same receptor, but often there are host-specific receptors, sorry, which means that the virus will use different receptor in insect and vertebrate. But these are data that are already very biased toward human and some vertebrates, and we don't have much knowledge about insects. So I think the big challenge for the future is to get more data on what's happening in insects with these viruses and study more what is really there, how is the virology there, which will help us actually to control this disease directly in the mosquito and insect. Also, I mentioned it, there are host barriers, which are different in insect and in vertebrates. So these viruses need to face the antiviral system of the vertebrate and the insect. So we have already a lot of knowledge on both, but not much knowledge about how these different viruses fight the antiviral system into the insect. Okay, so you have, for example, here a list of different genes that are impacted into resistance in insect for viruses, for acquisition, dissemination, transmission in this publication. So you see there's a lot already known, but how the virus bypass this is not well described. Also, could be interesting using bioinformatics and studies to look directly into the genome of the host, insect host. So I show you, for example, of course, there are gene bank, and there are a few databases that give you more tools, like, for example, Mercier Analytics in the US, which gives you analytics for pathogen, host, and vector data. So it's kind of nice. We are speaking about vectors. And for example, I type on their list some Aedes mosquitoes. So I got 2, Aedes aegypti and Albopictus. You see you have 2 and 3 assemblies. And then you can get all the different sequence, access to it, on chromosomes. And they can send the data to Galaxy. I told you the place where you have a mashup of different tools, bioinformatic tools. So you can select your genome of a mosquito, whatever, send it to work with tools and make some analysis on it if you want to. Okay,